So hi guys, today we are learning class 9, 5th chapter, the fundamental unit of life. So let's begin. Page number 59. First question. Who discovered cell and how? Answer. The cells were discovered in 1665 by an English botanist, Robert Hooke. He used a primitive microscope to observe cells in a Koch's scale. Second question. Why is cell called the structure of a functional unit of life? Answer. The cell consisted various compounds of plants and animals. A cell is the smallest unit of life and is capacity of all living functions. Cells are made the building block of life. This is the reason this is the reason why cells are referred to as the base structural and functional unit of life. All cells vary in their shapes, size, and activity. They perform, in fact, the shape and size of the cell is related to the specific function they perform. Page number 61. First question. How do substances like CO2 and water move in and out of the cell? Discuss. Answer. The cell membrane is a selective permeable and regulated the movement of substance in and out of the cell. Movement of CO2. CO2 is a product reducing cellular respiration. Therefore, it is present in high concentration inside the cell. This CO2 must be excreted out of the cell. In the cell's external environment, the concentration of CO2 is low as compared to that inside the cell. Therefore, according to the principle of diffusion, CO2 moves from one region to higher concentration inside the cell towards a region of lower concentration outside the cell. Similar. CO2 enter the cell by the process of diffusion when the concentration of CO2 inside the cell is low as compared to its surrounding. Movement of water. Water move from a region of high concentration to region of low concentration through the plasma membrane. The plasma membrane as a semi-permeable membrane, and this movement of water is known as osmosis. However, the movement of water across the plasma membrane of the cell is affected by the amount of substance dissolved in water. Question number 2. Why is the plasma membrane called a selectively permeable membrane? Answer. The cell membrane of plasma membrane is known as a selectively permeable membrane because it is regulated the movement of substance in and out of the cell. This means that the plasma membrane allows the entering of only some substance and prevent the movement of some other materials. Page number 63. First question. Fill in the gaps. The following table. Difference between prokaryotic and prokaryotic cells. Prokaryotic cells. Here are some diagrams. Diagrams are not that much important. You take a screenshot. So we go further. Okay. Here are also one diagram. Question number 1. Page number 65. Can you can you name the two organelles we have studied that called their own genetic material, and mitochondria and plastids are the two organelles that contain their own genetic material. Both the organelles have their own DNA and ribosomes. Question 2. In the organism of a cell is destructed due to some physical or chemical influence, what will happen? Answer. The cell is the smallest unit of life which is capable of all living function. In the cell organism of a cell is destroyed due to some physical and chemical influence, then the ability of cell to perform all living functions such as respiration, nutrition, secretion, etc. could be affected. Question 3. Why are lysosomes known as suicidal bag? Answer. This question was very important. This question asked in exam always. So understand it very carefully. Lysosomes are membrane bound vessels structure that contain powerful digestion enzyme. These enzymes are capable of breaking down and foreigner food particle or microbic entering the cell. Sometimes lysosome can cause self-destruction of a cell, but realizing this digestion enzyme within the cells, hence they are also known as a suicidal bag. Question 4. Why are the proteins senesced inside the cell? Answer. Ribosomes are the state of proteins. Ribosomes are very small structure from neither in a free state superintendent in the cytoplasm or attended to the surface of the reticulum, endoplasmic reticulum. They are composed of ribonucleic acid and proteins. Page number 67. First question. Make a comparison and write down way in which plant cells are different from animal cell. Answer. Animal cell and plant cell. Animal cell. Animal cells are generally small in size. Plant cells are usually large. Then animal cell. Cell wall is absent. Cell wall is present. Expect the protein. No animal cell processing plastid. Plastid, chloroplasm, and lysoplasm are present. Vacuoles are small in size. Vacuoles are large in size. Here are some diagrams of animal cell and plant cell. So we go through first animal cell, mitochondria, Golgi body, nucleus, ribosome, cytoplasm, lysosomes, cell membrane, or here also plant diagram, cytoplasm, Golgi apparatus, lysosomes, mitochondria, vacuoles. Okay, so you learn it. So we go further. Second question. How is a prokaryotic cell different from a prokaryotic cell? Answer. Prokaryotic cell and prokaryotic cell. Most prokaryotic cells are unicellular. Most prokaryotic cells are multicellular. Size of cell is generally small. Size of cell is generally large. Nucleus region is poorly defined due to the absence of a nucleus membrane or the cell lack true nucleus. Nucleus region is well defined and it's surrounded by a nucleus membrane or true nucleus bond by a nucleus. membrane in is present in the cell. It contains a single chromosome. It contains more than one chromosomes. Nucleus is absent. Nucleus is present. Membrane bound cell organelles such as plastid, mitochondria, Golgi apag, reticulum, Golgi apag, etc. are absent. Cell organelles such as mitochondria, plastids, endoplasmic reticulum, Golgi apag, lysosomes, etc. are present. Cell divide occur only by mitosis. Cell divided occurring by mitosis and mitosis. Prokaryotic cells are found in bacteria and blue-green algae. Prokaryotic cells are found in fungi, plants, and animals. Question 3. What could happen if the plastid membrane rupture or break down? Answer. If the plasma membrane of a cell is ruptured, then the cell will die. The plasma membrane regulated the movement of substance in and out of the cell by diffusion of mitosis. Thus, if the plasma membrane is ruptured, then the cell might out in containing contents. Page number 767. First question: What happened? What could happen to the life of a cell if there was no Golgi apparatus? Answer: If there was no Golgi apparatus, the cell then most active performance by Golgi apparatus will not taken place. First, membrane of Golgi apparatus are often connected to ER membrane. ER full form is endoplasm. Okay, in collection separate molecule and combining them to make more complex molecule. They packed in small vessels and are neither stored in the cell or sent out as per the required. Thus, if the Golgi apparatus is absent in the cell, then about process of storage, modification, and packaging of production will not be possible. Second, the formation of complex sugars from simple sugars will not be possible as this take place with the help of present in Golgi body. Third, the Golgi apparatus is evolved in the formation of lysosomes or peroxisome. Thus, if the Golgi body is absent in a cell, the sense of lysosome or perox will not be possible in the cell. Fifth question: Which organelle is known as the powerhouse of the cell and why? Answer: The mitochondria are known as the powerhouse of the cell. Mitochondria create energy for the cell, and the process of creating energy for the cell is known as the cellular separation. From chemical reaction evolved in cellular separation occur in the mitochondria. The energy required for various chemical activity needed for life is realized by the mitochondria in the form of ATP, adenosine tributary molecule. For this reason, mitochondria are known as the powerhouse of the cell. Sixth question: Where do platelets and proteins consist the cell membrane get synthesized? Answer. Platelets and proteins consist the cell membrane are synthesized in the endoplasmic reticulum. SCR, smooth endoplasmic reticulum, helps in manufacturing of plates, and RER, rough endoplasmic reticulum, as particle attach to this surface called ribosomes. These ribosomes are the state of protein since. Question 7. How does an amoeba contain its body? Answer. Amoeba containing its body through the process of endocytosis. This flexibility of the cell membrane able the cell to confuse the solid particle of food and other material from a external environment. Here are an image. You can see first food particle, then he goes slow, and then cell configuration food particle. Question 8. What is meiosis? Answer. The movement of water molecule from a region of high concentration to a region of low concentration through a selective permeable membrane is called meiosis. It is a special cause of diffusion where the mirror of water, for example, if a medium surround the cell has a higher water concentration than the cell, etc. If the solution is a dual solution, then the cell will gain water by meiosis. Movement of water inside the cell. Question 9. Carry out the following meiosis experiment. Take 4 peeled potato, and snow spoon each one out the make potato. Cup. One of these potato cups should be made from a boiling potato. Put each potato cup in a through container water. Now, A. Keep cup limited. B. Put in teaspoon sugar in cup B. And C. Keep put 1 teaspoon salt in cup C. Put 1 teaspoon sugar in boiled potato cup D. Keep this for 2 hours. Then observe the 4 potato and answer the following. Explain why water generate in holy proportion of B and C. Second, why is potato unnecessary for the experiment? Third, explain why water does not generate in hello or out portion of A and D. Answer. Here are also diagram of potato sugar experiment set up. First answer, water gain in whole portion of setup B and C because water entered the potato as a result of mitosis since the medium surrounding the solution has a concentration higher water concentration than the cell. Water move inside by mitosis, hence water gain in a whole lead portion of potato cup. Question 2. Answer. Potato A in experiment acts as a control setup. No water generated in the whole portion of potato A. Third answer. Water does not generate in the whole portion of a potato because the potato cup is empty. It is not a control setup in the experiment. Water is not able to enter potato D because the potato used there in boiled boiling denature the proteins present in the cell membrane and thus disturbs the cell membrane for mitosis. A semi-permeable membrane is required which are distributed in the case therefore of meiosis will not occur, hence water does not enter the boiled potato cup. So how it is video? Like, share and subscribe to my channel.